Good morning. Thank you very much. It's a pleasure to be here. It's not so much thinking outside of the box as having the right box in which to think. That's what I'm going to put as my motto for Registries in Context. I want to expand and say and give you some areas of data sources and methodologies which both complement and add to registries. And I'm going to. And I'm also, because there's a series of speakers this afternoon on registries, so I'm going to, without hopefully not be too repetitious, I'm going to go into the area of safety, which we being an NCEP centre of the EMEA, we're very heavily involved in looking at safety issues, and give you some methodologies which I don't think are covered. Oh, right, thanks. First of all, a disclaimer, we have a conflict of interest. We're a sponsor, we're one of the sponsors of this conference, and we provide patient registries for both public and private sponsors. And we are a collaboration of Oxford Epidemiology, which provides methodology, Dendrite Clinical Systems, which provides the technology, and has been in the registries business for about 18 years, and Chiltern, which is a medium-sized global CRO, which is able to provide logistical expertise in many countries throughout the world. However, given that conflict of interest, I'm going to try and broaden the subject to just beyond registries. So what I'm going to look at the need to be a little bit creative beyond registries and within registries, the range of data sources and methodologies, the role of statistical signal detection, focusing on the role of registries rapidly, and then some of the methodological and practical issues. Next slide, please. So this is the conventional hierarchy of evidence for internal validity of interventions. You need large, unbiased evidence, and as we've seen, it's large, unbiased evidence in relation to how the biases could potentially change the conclusions. And as we've seen from the previous speakers, from the previous speakers, where you have such big effects, you don't actually need randomization. There was when penicillin was given for pneumonia, there wasn't a clinical trial that was required. It was so evident that any potential biases from the methodology would not overcome the conclusions which arose, and therefore this is the traditional hierarchy. So these are my principles. If you don't like them, I have some others, or I can adapt them. And in safety, the hierarchy of evidence is actually less pyramidal. You have the randomized clinical trial, you have non-randomized trials, you have data, you have disproportionality analysis, you have epidemiological studies, you have case reports and non-clinical data, and you need data evidence integration to be able to put that all together, often with systematic review methodology and meta-analysis. And the hierarchy of evidence for efficacy for ODs will come from in different situations, depending on how secure you are about the mechanism of action and the magnitude of the benefits. If the benefits are huge, then the randomized clinical trial may not be necessary, and non-randomized clinical trials can be adequate. And you can also use comparative observational studies in which to look at benefits. So this can come from cohort and even registries, from case control studies, and being supported by descriptive studies, which can be, again, cohorts and registries, prevalence and surveys, time trends and ecological associations. So all of these sources can be brought to mind depending on the particular situation that you have to hand. So at approval, it's changing from approval to not does something work, but what's the benefit and risk, and that's really what everybody, as a clinicians, that's what we're used to, and the regulators, and they're bringing in risk-benefit analysis in a formalized way, using them, the front runner is the multi-criteria decision analysis, which is being used. But again, for orphan diseases, I don't think this will be a problem because as I say, where you have effects which are overwhelming, then the model will be largely dictated by that, and the sensitivity analysis which will be run won't be very different. But the kind of evidence that you need is clearly randomized clinical trials, non-randomized trials. You need prevalence studies to demonstrate that you really do have an orphan indication. That's not difficult, but it needs a systematic appraisal from using epidemiological sources. And in the risk management plan, you need to have natural history, which hopefully you will get from a registry. There are other sources from that, electronic health records in administrative databases in the US, but also healthcare databases in Europe. The epidemiology, the adverse events of the condition, and if you're not the first for that condition, then what are the drugs that are being used, and the adverse events of the competitor, which again can be maybe available from a registry if there's one already available, or spontaneous adverse events database. There are lots of reasons for limited. There's lots of limitations of post-marketing surveillance, and we've heard that you can get approval with 50 patients. Clearly, there's a bigger question mark on the safety of that. Again, the benefit risk will decide as to how important that safety concerns should be, and what kind of safety concerns need to arise in order to annul the benefits which exist. And the type of adverse reactions which can occur post-marketing can be very different to those which occur pre-marketing. So in the European Union, in July of next year, there will be the new pharmacovigilance legislation, which is good pharmacovigilance practice, which concentrates on benefit risk, and the new benefit risk tool will be the PSUR, but there will be important elements for signal detection and past studies. Past studies will be the mainstay of most applications as a condition of approval, and so these are the kind of data that you will need to have in the post-approval arena. And again, product registries are an important ingredient of that in the orphan area, but also not forgetting spontaneous events databases. The detection methods, where you have rare diseases, you have to go to spontaneous reports and to epidemiological study, cohort case control studies and registries, because the other methodologies will not provide you with adequate data. So looking at specifically on spontaneous events databases very quickly, disproportionality analysis is where you get the adverse reports from your drug versus other drugs, and one looks at the disproportionality for various adverse events for those reports on your drug versus the comparator drugs. And there are proportional reporting ratios and odds ratios which are used in order to make that comparison. And so I'll just show you a very quick example from the WHO Digibase, which has 6 million cases from over 90 countries on all marketed drugs. And this is looking at Fabrazyme versus Repлагil for Fabry's disease. And you can see that you can look at these. You can do these disproportionality analysis on the WHO ART terms in Digibase. The FDA has MEDRA terms, and you can see that certain signals appear to arise. And you can, so I've highlighted in red some of the signals in this column here, and some of them may be. Yeah. So you can see that some of them may be conventionally statistically significant here with the lower confidence level above 1. But however, we use a Bayesian approach as well in these analyses, and all of these show that, which reduces the false positive rate, and you can see that there's really no difference between these two drugs in the adverse side effects reported. Looking at Gaucher's, looking at these two drugs, we looked at WHO critical terms, about 390 terms of serious adverse events, and you can see that again, the proportional reporting ratio is 1.05, and the Bayesian, estimated Bayesian geometric mean is about 0.99, very close. So there's no difference, showing there's no difference between the signals here in safety. So these are some of the things that you can do. The uses of these external adverse events databases, which the company will not have access to, so you need to go beyond your company, strengthen and weaken signals of a new adverse drug reaction association with a drug, comparison of safety profiles of drugs within a class, exploration of possible drug-drug interactions, looking at innocent bystanders. They can be used in peri-approval risk management planning for new drug applications. Even before your drug is on the market, you can use these to get insights for the risk management plan and you can proactively search for problems not detected during the clinical development program looking at other agents which are already on the market for that particular disease. And they can be used in quantitative risk-benefit models. And you can look at competing drug safety profiles. So very briefly, moving over to patient registries, definitely this is the NSET in the methods guide of the NSET, which came out very recently. Registry should be considered a structure within which studies can be performed. It's a data source where entry is defined by diagnosis of a disease registry, or prescription of a drug, an exposure registry. It's really a type of prospective cohort study. And in terms of design issues, this is a rather old publication which shows that when you educate people, you get an increase in reporting rates. This is ADRs, and the reporting rates. They trained this was in the Kaiser Permanente in HMO in California, and after education, you saw that the adverse event rate increased. Now, what that means is that if you just have there's been a debate about whether it should be product registries or disease registries. Now, if you only have a product registry, what that will do here is that if you just have one product registry, A will go up in relation to B, and so you'll record a higher incidence rate. One way to protect yourself from that is to have a comparator group, whatever that may be, and we can talk about what the comparator groups may be. C will also go up, so A over B versus C over D will now be cancelled out, so there won't be, from the changes in the reporting system rather than the drug itself should not increase the relative incidence rate for adverse events. The other thing about having patients in cohorts before the drug is on the market is that if you have, first of all, clearly you have a lot more patient years of exposure to look at. Looking at the natural history here, so you have a lot more patient to look at the natural history, but also when your drug is introduced, then you also have a lot of comparative data on patients prior to, for example, an adverse drug reaction appearing. So having a cohort in well before your drug is approved will both help protect you from any adverse events, putting adverse events properly into quantitative context, and also allowing you to have a lot more data on the natural history. So the place of registries, where you have gaps in natural history, treatment and safety, and that's been adequately discussed today and yesterday. Where you want effectiveness and safety in real-world patients, particularly long-term and delayed benefits and hazards, you need registers for that. Where you're looking at diagnostic paths, the treatment pathways, the patterns and the variations. When you want to look at subgroups, and when you want additional outcomes, the registries have a place. There are clearly biases of observational design, but those should not be overwhelming if for registries to be in place. If they are, then a registry is not an appropriate design. And where there are those concerns, registries can also act as pragmatic trials, and we have examples using the systems that Denwright have been running for at least 18 years, in which there are examples of pragmatic trials through registry structures. So why invest in registries? Clearly fill data gaps, build a clinical community which will help increase patient recruitment, and also establish at the time of approval. Then when you go to agencies, health technologies agencies like NICE, you can use data from the registries in order to feed into the cost effectiveness models which you may where these have to be built. Also, it obviously helps launch. There are certain scientific challenges which I probably, depending on how hungry you are, we can go into or not. But clearly, one needs to have clear objectives. There can be the challenge of having too much data. Specialists like a lot of data. You really have to be ruthless in terms of what the key elements should be. What are the comparators? In terms of objectives, we like one of the way we try and discipline people is we try and relate the primary and secondary objectives to the variables for analysis and what the actual analysis will be, so that they can see exactly what this information will be, how it will be used, and what will be generated at the end of the study. Clearly, too much data. Focus on primary and secondary objectives. What kind of comparators should you have? Clearly, internal comparisons are the best, and they can be both with other patients that are not on the study, patients that are on other drugs, but also in terms of before and after comparison, the self-control comparison. However, if you don't have those, you can go to other registry data, you can go to historical data. There are historical data sets. For example, the Mayo Clinic has been looking at fairly rare diseases and has data on that. You can also go to electronic health data sets. For example, the INSEP in Europe is amalgamating, has about 30 million people in these databases, and for certain types of rare diseases, one can get data for comparison. The time windows for analysis are important in looking at comparisons. Attrition is always a problem, and how to collect ADRs. Well, you really need a good PV plan and good pharmacovigilance with it, and particularly there's pharmacovigilance is encroaching increasingly on the obligations of observational studies, both in the orphan disease area but elsewhere. So clearly issues about validity, precision, generalizability, all of this is study and disease specific. These are general rules and they have to be applied to each particular area. And these are some of the my chairman saying that I've got lunch is awaiting. So these are some of the registries that Denwright have been associated with over many years: Gaucher, cystic fibrosis, Guillain-Barré, MEN1 and MEN2. These are endocrine disorders. There's also NF2 registry. There's also sarcoidosis registry, which when I practicing doctor didn't think sarcoidosis was an orphan disease, but apparently it is. So the world moves on. I don't have time, so if anybody, the practical issues are based around recruitment and retention of both centres, and patients. And if people are interested, we have a booth that people can come to. So just to summarize, role of patient registers in rare disease is crucial. Many scientific and practical challenges exist. Each is different to the disease. Other data sources can complement registries, and evidence integration is important. Thank you very much.